Assalamualaikum and warm greetings from Terengganu. In the previous video, you have been introduced to general aspect of fish parasitology and their major groupings. In this topic, I would like to introduce to you about the parasitic protozoan that commonly infecting fishes. Some of the topics covered are listed here, such as the general introduction to protozoan, where this section will help you to understand the meaning of the group, some general morphology and important group to fish health. You will also be introduced to the mobility and locomotions of protozoan in this section. The following section will be specifically focusing on the flagellated protozoan followed by ciliated protozoan. In these two sections, I will provide a preview on some important species related to pathogenicity and diseases of fishes. Some aspect of their morphology and host selection will also be discussed throughout this course. So, let's start the course. In Greek terminology, protozoans mean first animal, as protozoa mean first and zoa means animal. Protozoan comprise a spectacular diversity of unicellular, eukaryotic organisms possessing organelles such as a membrane-bound nucleus, mitochondria, chloroplast, Golgi, etc. that can be found in the metazoan plants and animals. Given their rich biodiversity and the enormous number of parasitic protozoan on fishes, it is quite impossible to discuss all of them in this video. Rather, we will be focusing our discussion on the flagellated protozoan precisely on these three phylum: dinoflagellate, Parabasilia and Euglonozoa, and ciliated species from phylum Ciliophora. That has been a concern in fisheries and aquaculture industry. Having a various living habitats, protozoa have evolved to exhibit distinct movement depending on where they normally live. In general, motility or locomotion in protozoa is usually mediated by cilia, flagella, or cellular appendages adapted for propulsion, or amoeboid movement. Other modes of protozoa locomotion involve gliding movement in which no changes in body shapes are observed. With the help of cilia and flagella, protozoan can use the features to travel longer distance per se that can maximize the possibility of encountering food particles. An example to demonstrate the movement of ciliated or flagellated protozoan can be seen from the YouTube link provided here. In this video, you can observe the swirling movement that protozoan made from their cilia or flagella to move their body. Let's now begin with the first flagellates group from the phylum Dinoflagellates. Protozoa placed in this phylum were characterized by presence of one or more flagella. Flagella and cilia are permanent microtubular organelles that are anchored within the plasma membrane of certain protozoa and project from the cell surface. Many dinoflagellates produce ichthyotoxin, which has caused mass mortality in wild and cultured fish. About 140 of the approximately 2,000 known living species are parasites, most being parasites of invertebrates. These animals can be found commonly on the external parts of the fish, such as skins, fins, or gill, although some species might infect internal fish organs. They feed as sessile, sac-like trophozoites on the skins or gill epithelium. Within phylum Dinoflagellates, 5 genera has been reported as fish parasites, which are Amylodinium, Piscinodinium, Crepidodinium, Ichthyodinium, and Uudinoides. Only Amylodinium, Piscinodinium, and Ichthyodinium are of any known or potential economic significance. The first example of dinoflagellates is Amylodinium. Amylodinium is a warm water parasite and it is more pathogenic at higher temperature. Thus, in more temperate regions, it is only a problem in warmer months. The trophon, which is the mature developing stage of Amylodinium, is pear-shaped to avoid and can be measured up to 350 micrometer long. Amylodinium is one of the few fish parasites that can infest both elasmobranchs and teleos, and almost all fish that live within its ecological range are susceptible to infestation. Amylodinium has continued to be one of the most serious impediments to warm water mariculture, with well over 100 species known to be susceptible. In this slide, you can see the schematic diagram of host attachment mechanism of Amylodinium. The blue arrow here indicates the host cells such as the skin, the fins or the gill epithelium layer. At the basal regions of Amylodinium attachment, you can see numerous filiform rhizoids which is highlighted with red arrow. The rhizoids are enlarged at their bases, which serve to anchor them deep in the host cell. The second example of dinoflagellate here is called Piscinodinium pilulare. The trophons of Piscinodinium pilulare is a yellowish to greenish to brown in color, and they showed a piriform or sac-like cell. Mature stage can be measured up to 140 micrometer long. They are almost round when mature and appear to possess a rudimentary sulcus. Piscinodinium pilulare is considered as one of the highly pathogenic species. Many tropical fish are susceptible to Piscinodinium, with anabantids such as Siamese fighting fish, gauramis, cyprinids such as goldfish and barbs, and cyprinodontids such as killifish frequently affected. In this slide, you can see the schematic diagram of host attachment mechanism of Piscinodinium. Just like Amylodinium, Piscinodinium attachment also involved the insertion of rhizoid, in red circle and arrow here, into the host cell. However, unlike Amylodinium, the rhizosis of Piscinodiniums are thin, nail-like holdfasts that radiated from the stalk. The shaft of the rhizosis are firmly embedded in host cell cytoplasma. In this next flagellates group, we will discuss about parasites belonging to phylum Parabasalia and phylum Euglenozoa. Members of these two phylum are flagellated protozoan that are generally found in the digestive tract and blood body fluids of vertebrates such as fishes, amphibians, reptiles, birds, and mammals. In addition, some species are ectoparasitic and are found on the body surface and or gills of fishes. In the course of our topic, we will review mainly on genus Spironucleus, Ichthyobodo, and Cryptobia that are known to cause disease in freshwater and marine fishes. The next parasite is called Spironucleus from phylum Parabasalia. Shapes of the live Spironucleus tropozoites vary from elongated to nearly spherical with length up to 20 micrometer. Each side has a nucleus and 4 flagella. Flagella lengths are about 1.5 to 2 times the length of their body. Spironucleus are frequently reported in the digestive tract of fishes and sometimes also in internal organs and tissues. These parasites have direct life cycles, and some species are associated with morbidity and mortality, disease known as spironucleosis, and possibly hole-in-the-head disease in salmonids, cyprinids, and ornamental aquarium fishes. Another protozoan parasite that causing serious threat to fish is Ichthyobodo species. For your information, this parasite has a free living stage in water and a parasitic living stage which is usually attached to dorsal fin, the tips of gill lamella of fish, and also the mucosal layer of the body. The inserted figure here, pointed by the blue arrow, is the parasite while they are in free living modes, while the figure behind it is Ichthyobodo in their parasitic modes. Almost all fishes are susceptible to Ichthyobodo infection, and this parasite has a wide range of temperature adaptation. Some signs showed by the fish prior to infection is increasing in mucus production, scratching behavior, and often gasping for air. The next flagellate is Cryptobia species. Briefly, this parasite is elongated with 2 flagella which originate from the anterior end. The shorter anterior flagellum is free, while the recurrent flagellum is attached to the body and its ends as free as flagellum at the posterior end. Cryptobia species infect and cause diseases in many species of marine and freshwater fishes such as salmons and flatfish. The parasite may be on the body surface and or gill such as an ectoparasite, in the digestive tract, and in the blood vessel of the fish, and become endoparasite of the fish. Some of the pathogenesis of infection are necrosis of epithelial layer, increased mucus production, while signs of infections are gasping and emaciation. We have now know a little bit on the parasitic flagellates. Now, let us discuss yet to another group of parasitic protozoan, the ciliates. Ciliates belong to the phylum Ciliophora, and as their name indicates, the members of this phylum were ciliated during at least one stage of their life cycle, moving typically by beating of the cilia. Ciliates exhibit both asexual and sexual reproduction, that is, asexual reproduction by budding, binary and multiple fission, as well as sexual reproduction by conjugation, autogamy, and cryptogamy. The phylum Ciliophora currently contains 7,200 known species, but there are probably several thousand more that are as yet undiscovered. Ciliates are often found on dead or moribund fishes, and parasitic ciliates are not common. In the course of this topic, we will describe the three most virulent parasitic ciliates of fishes, which are Ictyopteris multifilis, Cryptocarens irritans, and Trichodyna species. The first examples of ciliate protozoans here is called Ictyopteris multifilis. Ictyopteris multifilis, also referred to as ick, and is one of the most pathogenic protozoan parasite of fishes and has been known to parasitize all freshwater fish species. At almost all temperature range, nevertheless, the outbreaks of Ictyopteris multifilis occur when conditions are favorable for rapid multiplications of the parasite. These include a suitable environmental condition and susceptible fish hosts. In detail, a mature Ictyopteris multifilis can be measured between 1 to 1.5 mm in diameter. As you can see from the figures shown here, Ictyopteris multifilis possess a macronucleus that looks like a horseshoe, and infected fish will show a sign of gasping with an erratic run or flashing behavior. The following example is Cryptocarens irritans. For your information, Cryptocarens irritans is a holotrichous ciliate that infects surface epithelia of marine fishes. The various forms of this parasite reach approximately the same size as their Ictyopteris counterparts. The figure on the far end here is infective stage of Cryptocarens irritans that is called pteron, that has a piriform shape with 25 to 60 micrometer in length. Within a pteron body, there are a lot of trophon that will leave the fish as a free-swimming tomon. Just like Ictyopteris multifilis, ambient temperatures are reported to influence the growth and development of Cryptocarens irritans. The last examples of ciliated parasites that I would like to share with you today is the Trichodyna species. Trichodyna is a perithric commensalistic and facultative parasite. They can be usually found at the gills and the skins of the host, such as amphibians and fishes. If you can look in the middle of these images, you can see a distinguishable characteristic of Trichodyna, which is the presence of the denticular ring or tooth-like internal cytoskeleton ring. Around the body of the Trichodyna species, it has a ring of cilia or hair-like projection encircling the body and oral cavity that were used for locomotion and feeding. The Trichodyna species have a direct life cycle, and they reproduce asexually by binary fission under optimal conditions of the host fish and sexually by conjugation. Infected fish usually show signs such as scratching or presence of gray-blue turbid layer on the skin. Heavy infections of Trichodyna has been associated with secondary bacterial infection to the host. To check on more details about the Trichodyna, I recommend you to check on the following YouTube link where you can view the movement and locomotion strategy of these parasites. Now we have reached the end of our topic. I hope you can learn a little enough on the measure of important parasitic protozoan of fishes. From this video, we have learned the basic biological perspective of virulent protozoan through the examples given. Thank you very much again for your attention and see you again in the next topic. Goodbye.